In the next series of videos for enzymes, we are studying about the concept of activation energy. But before I explain about the concept of activation energy, I want you to know the nature of the enzyme action, right? So whenever there is a substrate, it needs to get converted into a product. It is catalyzed by an enzyme. So here you can see a structural enzyme, right? It's a representation of an enzyme. So what happens? The substrate goes and fits into the crevices or pockets in the tertiary structure of the enzyme, and it fits snugly, and then the enzyme fits in correctly, right? It encloses the enzyme properly to form a transition state, which is known as enzyme substrate complex. Please remember, this transition state structure is unstable. It is an intermediate unstable structure. This is going to form again another intermediate structure known as enzyme product complex. Once the product is ready, all the bonds are broken or made, then the product is released and the active site is free to go back to form the to bind to another substrate. So this is how the nature of enzyme is. This is how the enzyme acts. So after we learn this, we are moving on to study the concept of activation energy, right? Activation energy is nothing but the energy required for the substrate to attain the transition state, right? So it is a very important concept and very important graph. So this graph is given in your NCERT. So the x-axis is represented by the progress of reaction. The y-axis represents the potential energy. So here you can see the substrate is getting converted into the product, right? So the substrate is at a higher energy level and product is at a lower energy level, right? This is what you can see from the graph. But you can see that there is a transition state. What is this transition state? Transition state is nothing but your enzyme substrate complex, like I just now explained. So to attain that enzyme substrate complex, without the enzyme or in the absence of the enzyme, the activation energy is very high. But in the presence of the enzyme, the activation energy reduces, right? You can see the red one is in the absence of the enzyme. The pink is in the presence of the enzyme. So the activation energy is reduced. So if the energy required is reduced, it is going to happen much faster, is it? So if it is going to take the path of activation energy without the enzyme, then it has to go a long way to attain the transition state and then has to come down. But here it is taking a short way, so it is going to happen faster. So just in the previous video we studied about carbonic anhydrase, which forms 6 lakh molecules per second with the enzyme. So you can go back and see that video also to understand how effective enzymes can be. So if the product is at a lower energy level than the substrate, the reaction is exothermic. That is, there is no need of supply of energy. In fact, the energy is released out as heat energy. Endothermic is nothing but energy requiring reaction. So this graph given in your textbook is a exothermic reaction. Why? Because the substrate energy is higher, the energy is liberated out and the product energy becomes lower. Right. So endothermic means the product energy will be at a higher level. So whether it is an exothermic or an endothermic reaction, substrate has to undergo a much higher state of energy, which is known as transition state. The difference in the average content of substrate from that of the transition state is called as activation energy. So like I told you, the energy that is required to attain the transition state is what is known as activation energy. So enzymes eventually bring down this energy barrier, making the transition from S to P very easy, right? That is what is the concept of activation energy, right? I hope you people understood this graph. We will learn about the factors affecting enzyme activity in the next video. Thank you. Please subscribe to Chris Biology.